Video. Oh well. Go ahead and watch this short video about cell division.
Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoy. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of the cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and becomes short and thick to form the chromosomes. In metaphase, microtubules of the spindle interact with the chromosomes. In anaphase, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubule becomes shorter. In telophase, daughter chromosomes arrive at the pole and chromatin starts to decoy, followed by the reformation of nucleoli and nuclei. Where does Cal Cunningham stand on funding research?
Okay.
Give that bill on the floor tonight.
don't know what kind of accent that was. It's a little hard to understand some of the words she said, but the animation is pretty good. Video part. So let's look at chapter 7. Chapter 6 was about cell division, but it was about mitotic cell division, mitosis. And that's how most of the cells in our body divide to make new cells for growth and healing and replacing cells that die. In this chapter 7, we're going to learn about a slightly different kind of cell division that occurs only in the reproductive organs, the testes in male and the ovaries in females, for making sperm and egg. And it's called meiotic cell division, or meiosis. Okay, first some more terms we have to get familiar with. Diploid and haploid. You already know that a cell has 2 sets of 23 chromosomes, or 23 pairs of chromosomes, and the word for that is diploid. That just means that a cell that is diploid has 2 sets of chromosomes. A cell that's haploid has only 1 set of chromosomes. So a human cell that's haploid, instead of having 23 pairs of chromosomes, it has just 23 chromosomes. And the only human cells that are like that are the sperm and egg. They each have just 1 set of chromosomes. All the other cells in your body have pairs of chromosomes. For gamete, it just means sperm or egg. So the gametes are the sperm and egg. And they're both haploid. So a haploid sperm fertilizes a haploid egg. Now that fertilized egg has two sets of chromosomes, or 23 pairs, just like all other cells. This word somatic, a somatic cell just means any cell that's not a reproductive cell, not sperm or egg. And we've already seen that that when a cell is dividing, each chromosome has been replicated, it's been copied. So there are 2 sister chromatids still stuck together until they separate during the cell division. And one goes in one daughter cell, the other goes in the other daughter cell. But this word homologous, I don't think we've used this word before in this class, but chromosomes are in pairs. So you have 2 number 1 chromosomes, 2 number 2 chromosomes, 2 number 3 chromosomes, and so on. And those pairs are just called homologous. So if these would be the 2 number 1 chromosomes, we would call them homologous, a homologous pair. And the 2 number 1 chromosomes have the same kind of genes on them. So if, for example, on this number 1 chromosome, there's a gene for eye color right here, then the other number 1 chromosome would have a gene for eye color in the same place. It might not be the same color. One might be brown eyes and one might be for blue eyes, but they would both be the same kind of gene. Okay, reproduction in some organisms is sexual, and in some organisms it's asexual. The A prefix A on a word means not or without. So asexual reproduction just means reproduction without any sex going on. So with sexual reproduction, that just means there's a sperm and egg involved, a sperm fertilizing an egg. But in asexual reproduction, there's no sperm or egg. It happens some other way. Most one-celled organisms reproduce asexually. So when a bacterium reproduces, it just splits in half. There's no sperm or egg involved. So that would be a type of asexual reproduction. But many plants and even some animals can reproduce asexually. There are some animals that if the animal if the animal breaks in half, each half will grow into a new individual. That would be a type of asexual reproduction. Some animals, like sponges, but a sponge is actually an animal, and a sponge will, and some other lower animals like that will just grow a little, what we would call a bud. So if this was a sponge in the ocean, and it would grow a bud, and that bud would fall off and grow into a new sponge. There's no sperm or egg involved there. That would be a type of asexual reproduction. If you have a house plant and you want to have another house plant, you could cut a piece of that house plant on and plant it in soil, and it might grow into a new plant. That would be a type of asexual reproduction, making a new individual, but there's no sperm or egg, no sex involved there. With sexual reproduction, every generation is different because the offspring, the children have a combination of the genes from the father and the mother. That's why if you have kids, your kids aren't going to be exactly like you. They're going to have some of your genes and some of the other parents' genes. But in asexual reproduction, the offspring are just like the parents, unless there's some kind of genetic mutation happens during the reproduction. So we talked about the mistakes that happen when, for example, when a cell is getting ready to divide, all the chromosomes are copied, so there will be enough for both daughter cells. Cells make mistakes when they copy chromosomes. That would be a mutation. So if that happens, then one or both of the daughter cells might be genetically different from the parent. So that's what the only genetic variation is from mutations. If there's no mutations, then the daughter cells would be the same as the parents. If you take a cutting of a house plant and plant it and make a new plant, that new plant is genetically identical to the original plant. If this happens, a bud forms off and breaks off. That bud is going to be genetically the same as the original organism. Okay, life cycles. Life cycle is just everything that happens within the life of an organism, from the time that it's formed until the time that it dies or produces the next generation. Binary fission is the type of reproduction that one-celled organisms do. They just divide in half. Binary means two, fission means to split apart. So this is what most one-cell organisms reproduce this way, split in half. But sexual reproduction requires both parents, sperm and egg. The sperm are produced by meiosis or meiotic cell division, and then the mitosis is just for growth to become an adult. Okay, 3 kinds of life cycles in eukaryotic organisms, like plants, animals, fungi. There's some of them have a haploid life cycle, some have a diploid life cycle, and this alternation of generations, this is a combination of both. This is what plants do. To alternate means to take turns. So in plants, there's a haploid life cycle, and then a diploid life cycle, and then a haploid life cycle, and then a diploid life cycle. And we'll look at briefly what that means. In a haploid life cycle, fungi, an example of fungi would be bread mold, common kind of black mold that grows on bread. The cells of the fungus are actually haploid. They have just one set of chromosomes in them. Do we have a diagram of this? I forget. Yeah. These hyphae, this word hyphae just means the little threads that make up the mold. If you look at a mold under a microscope, it just looks like a bunch of little threads, and that's what they're called hyphae. And so these are the individual cells. And so those are haploid, and they do reproduce sexually, but they because they are already haploid, they produce haploid sperm and egg, not by meiosis but mitosis. So to go from a haploid organism to a haploid sperm and egg, the parent cells already have just one set of chromosomes, so there's no reduction in half of the chromosomes to make the sperm. So it happens by mitotic cell division. So you should have looked at this before class. This new textbook has new diagrams I'm not familiar with. In fungi, they don't call them male and female. They call them positive mating type and negative mating type. But it means kind of the same thing, except it's, they don't call them male and female because they don't have X and Y chromosomes like animal cells and plant cells do. But the only diploid part of the whole life cycle is this. It's called a zygospore, and so when the sperm and egg come together, they form this diploid zygospore. But it doesn't grow into an adult. It immediately undergoes meiosis to reduce the number of chromosomes in half. Forms these spores, which are haploid, that grow into the haploid adults, which produce haploid sperm. So it still involves sperm and egg, just like in in humans, but the adults are haploid. The cells of the adult fungus have one set of chromosomes instead of two. Where am I? Animals like us have a diploid life cycle. The diploid adults, that's what you are. You're a diploid adult because you're an adult and your cells have two sets of chromosomes, the diploid. But you when you produce gametes, sperm or egg, they're haploid. It's one set of chromosomes, and then they fuse together to form a diploid fertilized egg that grows into a diploid adult. So the adults are diploid. They produce sperm and egg, which are haploid. They come together to form a diploid fertilized egg. The diploid fertilized egg. This word zygote just means a fertilized egg, and then that grows by a lot of mitotic cell divisions into an adult. So this will divide in half by mitotic cell division. Two cells. Each of those will divide, and there's 4. Each of those divides, now there's 8 and 16, 32, and until you end up with trillions of cells. Many different kinds of cells in the adult. This third one, this alternation of generation, in the life cycle, there's an alternation or taking turns. There's a multicellular haploid organism, and then a diploid organism, and then a haploid one. What does that mean, or what does that look like? There's 4 different groups of plants: the mosses, the ferns, the cone-bearing plants like pine trees that produce cones, and the 4th group is the flowering plants. Most plants are 95% of plant species are flowering plants. So most of the plants we see out here are flowering plants. But they all have this alternation of generations kind of life cycle. But in the 4 different groups, the generations, these 2 kinds of generations look different. They look way different in mosses compared to flowering plants. But in a typical plant like a fern plant, that fern plant is diploid, 2 sets of chromosomes, and that's called a sporophyte generation. It's called a diploid sporophyte generation. The word sporophyte means spore-producing plant, the plant that makes spores. And so this adult fern plant makes spores, and this is it's a magnified view of the underneath of the leaves. If you look under the leaves of ferns, you'll probably see a bunch of little dots. That's where the spores are made. And spores are not like sperm and egg. A spore is just a small cell that can grow into an adult. That's really all it is. But these the spores are made by meiosis, so now these spores are haploid, 1 set of chromosomes, and they'll grow into an adult plant that is haploid. The cells have just 1 set of chromosomes. In a fern, this haploid adult, it just looks like a little green thing laying on the ground, you would hardly even notice it. You might just think it's a piece of a leaf that fell off a tree, but it's a little thing, maybe an inch, half an inch or an inch in diameter, laying right on the ground. But that's actually an adult fern plant. But it doesn't look like this. This is a diploid sporophyte generation. This is called the haploid gametophyte generation. Gametophyte means gamete-producing plant. It produces gametes, sperm and egg. So this little fern plant, it produces eggs and sperm. So the sperm will make their way over to the egg, fertilize it, and produce a diploid zygote that grows into a diploid sporophyte. So all plants are like this, have two different generations. And in the fern, the generations look like this. But in a flowering plant, like a daisy plant or a pine tree, or not a pine tree, but a maple tree, what you see, the plant that you see is the diploid sporophyte. They're the large plant that you see, but in those flowering, this haploid gametophyte generation is microscopically small. It's in fact there's a male gametophyte and a female gametophyte. The male gametophyte is so small it fits inside of a pollen grain. That's what a pollen grain is. It has a little tiny adult plant inside of it, but it's microscopically small. And the female gametophyte is called the ovule, and that stays on the plant. And in a flower, the ovule stays in the flower. So the pollen grain with a male gametophyte in it will either be carried by insects or blown by wind, and it'll land on a flower with a female gametophyte in it. And the little tiny adult plant in that pollen grain will produce sperm, gametes, and the ovule that stays in the flower will produce eggs, and the sperm from the pollen grain will fertilize the eggs in the ovule and form a fertilized egg, and that will become a seed. It'll be a little tiny embryo with a seed coat around it. It'll become a seed, and then that'll grow into a new adult sporophyte plant. So even though that pollen grain is very, very tiny, there's an adult plant in there that produces sperm, and something similar, a female version of that, that stays in the flower and produces eggs. So there's this extra step in the life cycle of a plant. In animals like us, we are diploid adults, kind of like this, except instead of having this whole extra generation, we just go right from, by meiosis, we produce sperm and egg, and then we're back to the diploid fertilized egg. We skip over this haploid generation. Towards spermatogenesis and oogenesis. Genesis, in a word, this means the origin of something or the formation of something, as in Genesis of the Bible. It's the origin of the heavens and the earth. So spermatogenesis just means making sperm. Oogenesis means making eggs. So this happens in animals. This happens in the testes and oogenesis, making eggs, happens in the ovaries. But as we saw, plants make sperm and egg also. Again, that life cycle, that haploid life cycle that fungi do. So the adult is haploid. Some organisms can do both a sexual and an asexual reproduction. In animals or organisms that can do that, when the conditions are favorable for the organism, everything's just going fine, they will reproduce asexually. And if the conditions are good for that kind of organism, then it would be just fine if the next generation would be the same as the present generation. So why change? If the organism is well adapted to the conditions, then the next generation will also be well adapted, so they'll be the same. But if the environmental conditions change, start to get warmer or colder or drier or wetter, so that it's no longer favorable for that kind of organism, then it might switch to sexual reproduction. Because with sexual reproduction, there will be a recombining. There'll be combining of genes from the mother and the father, so that the next generation would be different. And that might be good. If the environment's changing, then the organisms have to change as well to adapt. So it might be a good thing to switch to sexual reproduction, so there would be more variation, more generation more genetic variation from generation to generation, so that at least some of those individuals in the next generation might be better adapted to the changing conditions. Okay, again, more about alternation of generations. Just another slightly different diagram saying the same thing. Diploid sporophyte, adult plant produces haploid, it's haploid cells, but these will be spores that will grow into a haploid adult. This gametophyte that produces gametes, sperm and egg, and then fertilization, the fusion of the sperm and egg produces a diploid fertilized egg that grows into a new diploid sporophyte adult. That's very confusing. I learned this in general biology class. Never really understood it. And I took a few botany courses, courses on plants. And I learned this several times before it really sunk in, and I really understood it, and it made sense to me. Okay. The bottom of these slides is cut off, so well, there's nothing there. I guess this, I didn't make this question. It says compare and contrast asexual and sexual reproduction. You guys know what a Venn diagram is? You ever learned what a Venn diagram is? It's shown as two overlapping circles. So if you were going to compare and contrast two things like asexual and sexual reproduction, you would put the characteristics of or description of asexual reproduction here, and the description of sexual reproduction here, but in here where they overlap, you would put what they have in common. Um, what sexual and asexual reproduction have in common here would be in that overlapping area. And so there's what they have in common is in reproduction, either way, it's producing new individuals, obviously. Genetic material passed on from parents to the offspring, but in asexual, there's only one parent, in sexual, there's only two parents, so those are different, so you don't put that in the overlapping part. Asexual offspring are genetically identical to the parents unless there's a genetic mutation occurs during the reproduction. But in sexual reproduction, the offspring are typically not genetically identical to the parents because there'll be some combination of some genes from one parent, some genes from the other. Mitotics or mitosis cell division versus meiosis. No gamete, no sperm and egg involved in asexual, but they are in sexual. Okay, some more practice questions. Okay, meiosis. Keep talking about this, but what exactly is it? It's meiosis or meiotic cell division. So we learned about mitotic cell division in chapter 6. That's called duplication division because the daughter cells are the same as the parent cell. But meiotic cell division is called reduction division because this kind of cell division results in daughter cells that have half as many chromosomes as the parent cell. The parent cell is diploid. The daughter cells, which will be sperm or egg, are haploid, have just one set of chromosomes. And this involves two cell divisions, one after the other. And those divisions are called meiosis 1 division and meiosis 2 division. And there's the time in between those two divisions is called interkinesis. And the reason, because there's two divisions, two divisions. So in the person's, for example, a person's testes or ovaries, there's a parent cell that divides into two daughter cells, and then if each of those would divide, then there would be 4 daughter cells from this meiotic cell division. If this happened in a man's testes, then each one of these would grow a tail and become a sperm cell. And these are, this is diploid, 2 sets of chromosomes, but each one of these is haploid, 1 set of chromosomes. Shorthand for diploid is 2N, and haploid is N, where N is how many chromosomes are in 1 set. So in humans, N is 23. So each diploid human cell has 2 sets of 23 chromosomes. Other species have different numbers of chromosomes. So here's a summary of meiosis cell division. And then in this diagram, this parent cell has just 2 sets of chromosomes, 2 pairs of chromosomes. If this was humans, that would be 23. But to make it simpler, this diagram just has 2 pairs. So there's a long pair and a short pair. So let's call the long ones number 1 chromosomes and then the short ones number 2 chromosomes. And each pair has a red one and a blue one. And so, this person had two parents, and the one chromosome came from the mother and one from the other, from the father. Um, so you have 23 pairs of chromosomes. Each of those pairs of chromosomes includes the one that came from your father and one from your mother. So just like before mitotic cell division, when a cell's getting ready to divide, it replicates this DNA. So it makes a copy of every one of these chromosomes. So now there's these same two pairs of chromosomes, but now there's two sister chromatids stuck together for each one. There's two copies still stuck together, just like before mitotic cell division. Then in meiosis 1, the first division, this is different from in mitosis, where the sister chromatids separated into the daughter cells. In meiosis 1, the two chromosomes of each pair separate into the daughter cells. But the sister chromatids of each chromosome are still stuck together in these two daughter cells. But now there's only, after meiosis 1, there's only one pair of chromosomes, or one set of chromosomes. They're not pairs anymore. So there's one number 1 chromosome and one number 2 chromosome in each of these daughter cells. But they're still replicated. Now in meiosis 2, what happens is similar to mitosis, like cell division, where the sister chromatids finally separate into the two daughter cells. In each of these divisions, meiosis 1 and meiosis 2, there's a prophase, metaphase, anaphase, and telophase, similar to what happens in mitotic cell division. Okay, something that happens in meiosis cell division that doesn't happen in mitosis is during meiosis 1, the two chromosomes of every pair, they come together side by side, and that's called synapsis. So this can be the two number 1 chromosomes will just come together side by side and form a tetrad. So we call this a tetrad. A tetrad means just 4 of something. So there's 1, 2, 3, 4 sister chromatids in this tetrad. And while they're together like this can happen. It's called crossing over, where the parts of these two chromosomes of a pair can break and swap between these two chromosomes. This should only happen if these are both number 1 chromosomes. This should not happen if this is a number 1 chromosome and this is a number 2 chromosome. This should only happen in pairs. But now these two sister chromatids are now different. But before, these were identical. And these were identical to each other. But now these sister chromatids they're different. They're different from this one, different from this one. So there's two whole new sister chromatids here that have a different set of genes. This has some blue genes and some red genes. So we call these recombinant chromosomes, because they have a combination of genes from the two original chromosomes. So this is called crossing over, and this is one reason. So this happens while you're making sperm and egg, this crossing over, and this is one reason why every sperm is different. Every egg is different from a person, because this happens all the time, and it could happen anywhere along these chromosomes. It could happen here, or here, or here, or here, or it could happen more than one place. So there could be a red section and a blue section and red and blue all on one sister chromatids. And because it can happen anywhere, there's an infinite number of possible chromosomes that one person could make in the sperm or eggs that the person produces. Another reason why every sperm and egg is different is it's what we see happening here. When these chromosomes of each pair, when they separate in meiosis 1 and go into these daughter cells, the way it happened in this diagram is this daughter cell got a blue number 1 chromosome and a red number 2 chromosome, and this one got a blue number 2 and a red number 1. But it could have happened a different way. It could have been, this daughter cell could have gotten a red number 1 and a red number 2, and this one got a blue number 1 and a blue number 2. Or this one could have gotten a blue number 1, a blue number 2, and a red number 1, and so on. So with just 2 pairs of chromosomes, there's 4 different possibilities of combinations of chromosomes that could end up in these daughter cells, and then in the final daughter cells after meiosis 2. So that's just with 2, not counting crossing over. Just the fact that any combination of these chromosomes could end up in these daughter cells, that could result in 4 possible different sperm or eggs that one person could produce. But because in humans there's 23 pairs, there's something like 70 trillion different combinations of red and blue, one from each pair. Something like 70 trillion combinations of different sperm or egg that one person could produce. And that's, again, why every child you have is going to be different. Because every sperm and egg you make different, and every sperm and egg that the other parent makes is going to be different. And that's called independent assortment of chromosomes. That just means that the red ones don't all go in this one, and the blue ones all go in this one. Which chromosome of each pair that goes into a particular daughter cell is independent of which chromosome of the other 22 pairs goes in to each daughter cell. So it could be any combination. This is a crossing over again. Again, in sexual reproduction, there's a lot of genetic variation from generation to generation. Your kids don't look like you; they're different because there's a different combination, and that genetic variation is because of crossing over during meiosis and independent assortment of chromosomes during meiosis. So that just means that every sperm and egg you make is going to be different. So there's an infinite number of different combinations of genes from your chromosomes that could end up in each sperm or egg that you produce. Another reason for genetic variation is that within a species, any sperm can fertilize any egg. This is showing what I was saying. Just showing the different combinations of chromosomes that can result from independent assortment. So again, we start out with the parent cell with just two pairs of chromosomes, a red and a blue of each one, and you could end up with these four possible combinations in sperm or egg, either blue and red. Hardly see it. I'm colorblind. I can hardly see that. Is that blue and red, blue and blue, red and red? I don't know. But there's four different combinations from just two pairs. If this was 23 pairs, it's something 70 trillion combinations of chromosomes that are possible. Okay, so here's more detail of the two divisions of meiosis, showing the different phases and what happens. And it's the same kind of thing. Prophase, same thing happens. Nuclear envelope goes away. The chromosomes coil up, or condense and become thick and short. And then the mitotic spindle with the protein fibers form, and they move the chromosomes so they line up in the center. And then they start to pull apart, anaphase, and then the two sets of chromosomes are completely separated, and then the two daughter cells form, and the whole divides in half. But how this is different from mitotic cell division is when these chromosomes line up in the center in metaphase, the chromosomes of the pairs are side by side. In mitosis, they're just by themselves, all lined up. But the synapses happens, so both number one chromosomes are side by side. Both number two chromosomes are side by side in metaphase of meiosis one. And then when in anaphase, it's the chromosomes of a pair are pulled apart, not the sister chromatids of each chromosome. They stay together. And only in meiosis two, that's when the sister chromatids of each chromosome separate into the daughter cells. And so if I said that this is what would happen to produce sperm, so each of these daughter cells would become a sperm cell. It's a little different in females and humans. Because after meiosis one, this first division, there's two daughter cells, but one of these daughter cells, it just dies. And this will be this daughter cell that dies. It's very small, very small daughter cell, and it just dies. And this is called a polar body. This daughter cell that dies, and so no second division for that one. It just dies. And then when this second division, when it happens, there's one little tiny one, and one little tiny daughter cell, and one full-size one, and this one dies. And so this is called the first polar body, and this is called the second polar body. So in females, in the ovaries of a human female that are producing eggs, there will be only one egg from the original parent cell. The other daughter cells just die. Okay, so here's a list of differences between meiosis and mitosis. So meiosis, 2 divisions. Mitosis was just 1. Nuclear division means division of the nucleus and the chromosomes. Meiosis produces 4 daughter cells. Mitosis produces only 2 daughter cells, but in the case of females, there's only 1 daughter cell from meiosis to make eggs. In meiosis, the daughter cells are haploid, but in mitosis, the daughter cells are diploid, just like the parent cells. After meiosis, the daughter cells are genetically dissimilar. That means they're different from each other and different from the parent cell, but in mitosis, they're the same. Meiosis, there's a synapsis and tetrads form and then crossing over. None of that happens in mitotic cell division. And like I was saying a minute ago, in metaphase 1 of meiosis, metaphase 1 just means metaphase of meiosis 1 division. That's where we saw that it says the tetrads align at the equator. That just means that the pairs of chromosomes are side by side during metaphase. But that doesn't happen in mitosis. Like we saw in anaphase 1 of meiosis, the homologs, or the chromosomes of each pair, separate, and then the sister chromatids don't separate until meiosis 2. Whereas in anaphase of mitosis, the sister chromatids separate. Okay. So here's a comparison of meiosis. So this is meiosis. So meiosis 1 division, and then this meiosis 2 division, and this is mitosis down here. So here we see in meiosis 1, in metaphase, where the chromosomes all line up in the center, the pairs are side by side. So number 1 chromosomes are together, number 2 chromosomes are together. If this was a human, all 23 pairs would be paired up like that during metaphase. And then in anaphase, the chromosomes of each pair separate into the daughter cells. And it's not until meiosis 2 that the sister chromatids of each chromosome separate into the 2 daughter cells, which is what happens in mitosis. Mitosis is the individual chromosomes line up in the middle. They're not in pairs like this. You just line up, and then the sister chromatids pull apart into the daughter cells. Okay, some practice questions. Chromosomal abnormalities. How far are we here? Keep going a little bit more. Okay. This arrangement of the chromosomes of a cell is called a karyotype. It's a karyotype. So they don't look like this in a cell. They're not arranged like this, but a scientist in a lab has done this. Took somebody's cell, took the chromosomes and paired them up like this, and the chromosomes are numbered from longest to shortest. So this was a human cell with 23 pairs of chromosomes. The number 1 chromosomes here, this pair, those are the longest, and then they get shorter and shorter. So the number 2 chromosomes are pretty short, and then there's this pair here called X and Y. Those are called the sex chromosomes. X and Y chromosomes. So that's that would be the number 23 chromosomes, and we just call them the sex chromosomes. All the others, we call them autosomes or autosomal chromosomes. And this individual has an X chromosome and a Y chromosome. Would that be a male or female? Anybody know? Yeah, male. Males have X and Y, one X and one Y. So what do females have? Two X's, two X chromosomes, right? So females don't have a Y chromosome. So I already mentioned that mistakes can happen during the cell cycle. When chromosomes are being copied in the S phase or interphase, when the cell's getting ready to divide, the cell can make mistakes. And so that one of the sister chromatids will not be exactly the same as the other. They're supposed to be identical, but if the cell makes a mistake, that would be a mutation. Sometimes mistakes happen during meiosis when the chromosomes are supposed to separate into the daughter cells, or the sister chromatids are supposed to separate into the daughter cells. What if they don't separate right? That's a mistake that happens sometimes. So we end up with some of these abnormalities, or chromosome disorders, or chromosome anomalies, they're sometimes called. We end up with the wrong number of chromosomes in a person. That happens sometimes. This term euploid means an individual with the correct number of chromosomes. Like most of us probably have the normal 23 pairs. This prefix EU means true. So if you have the true correct number of chromosomes, so you're euploid. Non-disjunction is a mistake that happens during meiosis sometimes. So the disjunction, junction means connection. So disjunction would mean disconnect, which is what happens during meiosis. The sister chromatids disconnect and go into the daughter cells, or the chromosomes of each pair separate, disconnect, and go into the daughter cells. Non-disjunction would mean it doesn't happen like it's supposed to. So what if instead of the sister chromatids separating into the daughter cells, maybe the number 7 chromosomes, they both go into, or both sister chromatids go into one daughter cell. The other daughter cell doesn't get any number 7 chromosome. That would be a case of non-disjunction. And so that can result in polyploid. Poly means many. But 3 or more complete sets of chromosomes in one individual. Normally in plants and animals, there's 2 sets. But in some cases, an individual can end up with 3 sets of chromosomes. And that's not uncommon in plants. I don't know if it's even possible for a person to be alive, to survive, and have 3 full sets of chromosomes. I don't think that's possible. So the term aneuploid, too few or too many of one type of chromosome. So if you had an extra number 7 chromosome, that would be a case of aneuploid. You have too many of one type, too many of one of the chromosomes. Or if you were missing a number 7 chromosome, you probably wouldn't be alive if you were missing a whole chromosome like that. But that would be a case of aneuploid. So a lot of these cases of aneuploid just result in a miscarriage. That person can't even develop and to be born. Would die very early in pregnancy. Monosomy is a case of aneuploid in which there's only one of a type of chromosome. So instead of 2 number 7 chromosomes, you have only one. And trisomy, cut off here, but trisomy, tri means 3, so it means you have, like, if you have 3 number 7 chromosomes instead of 2, we would say that we would call that trisomy 7, 3 number 7 chromosomes. Some people have extra X chromosomes or extra Y chromosomes. So some people are like this, 3 X's, or some people are like this, or I think there's some people like this, X, Y, Y. There's no such thing as a living person who's like this, Y, Y. So if this happened by accident, this would be a miscarriage. This person wouldn't survive. There's too much important information on the X chromosome. You have to have it to survive. In the past, I put an extra credit question on exams. Like, is it possible? Or name a person who has no Y chromosome. And some people, many of the students just leave that blank. And when any female, just name any female, that's the answer. Every female has no Y chromosome. Yet people are confused when I ask. It's so simple that they don't get it. Yeah, every female does just fine without a Y chromosome, but no one can live without an X chromosome. So here's some cases of non-disjunction. So what's going on? The non-disjunction during meiosis 1, when the two pairs of the chromosome pairs are supposed to separate, one from each pair goes into each daughter cell, but in this case, the long chromosomes, they didn't separate like they're supposed to. They both went into this daughter cell. This daughter cell didn't get any, and then after meiosis 2, end up with this daughter cell has two of the big chromosomes, one small one, 2 big ones, 1 small one. This one just has a small one, not any of the big ones. So this is monosomy. This would be monosomy. This would be trisomy, I guess. Non-disjunction can happen during meiosis 2. So meiosis 1 happens just fine, but then the sister chromatids don't separate like they're supposed to. Like, these 2 sister chromatids are still stuck together in this final daughter cell. And the other daughter cell, you can get one. So that's a mistake. So these would be sperm or egg here. So if this sperm would fertilize an egg, now that fertilized egg is going to be abnormal. It's going to be missing a chromosome or have too many chromosomes. So Down syndrome most cases of Down syndrome are caused by trisomy 21, a third chromosome, number 21. And so one of the reasons for doing a karyotype like this, why would you bother doing this for somebody? Because if you did a karyotype, if this was a karyotype of cells taken from a fetus. And you would do a karyotype, and you would see that there's 321 chromosomes, number 21 chromosomes. That when that baby is born, it's going to have Down syndrome. So, so you can tell from a karyotype, you can tell if there's missing chromosomes or extra chromosomes, and that's information that a parent might want to know about before the baby is born. Oops. Some others, Turner syndrome, just one X chromosome. This would be a female. She has just one X chromosome, not two. Klinefelter's, this would be a man with an extra, at least one extra chromosome. So XXY is called Klinefelter. These people can survive, and there might be some people who have that one of those, and they're, they look a little different. People, I think, with Turner syndrome, these females, they're usually small size, and they look childish, have childish looking faces, and they're usually sterile. They can't reproduce. But you might know somebody like that, and you would know that she has that. Klinefelter, these are men who are usually tall and have long, slender arms and fingers, and they have kind of wide hips and narrow chest. Look kind of effeminate. Don't look very masculine. But I think some people say that George Washington might have been Klinefelter, just from pictures of him and the fact that he and Martha never had kids. I think it's too late to dig him up and count his chromosomes. Okay, just several others of these chromosomal abnormalities that are common enough that they have names. Down syndrome, Tall syndrome, Edwards, Turner syndrome. The word syndrome just means a syndrome. We use the word syndrome instead of the word disease sometimes. Syndrome is just a collection of symptoms, or a collection of just abnormal characteristics that occur together in people, and so we call it a syndrome. In some cases, we don't even know what causes it, but it happens often enough that we give it a name, call it a syndrome. Like sudden infant death syndrome. For a long time, no idea why it happened. Some babies just died, so they call it sudden infant death syndrome. Okay, we need to take a break, I guess. So let's take a break. Finish this up and make it for 8 a little bit. have a thermometer in here now. Anybody want temperature taken? Just use the forehead, right? 36.3 Celsius. How do I make it Fahrenheit? Oh, that's the room. The room. Who's here? Haley, Kamani, Priya, Sharika, Cameron, and Tyson. Was it Tyson? Somebody, who was that? Was that Jacob or Rylan? You know? Jacob, he was here. I don't know if he's coming back. Class is shrinking. I know this stuff is boring, but we got to learn it. Okay, almost done with this chapter. So we're talking about these chromosome abnormalities of the wrong number or wrong quantity of one or more chromosomes. But there are also chromosome structure abnormalities. So this is an abnormality within one chromosome, something wrong. And there are several different types of these. A deletion, to delete something means to remove it. So there can be part of a chromosome missing. So maybe the end of a chromosome is missing, it just broke off. Or in some cases, a chromosome broke in two places, and then the in-between piece is missing. So this piece is missing, and the two ends came back together. So that would also be a deletion. Duplication. Part of a chromosome is accidentally duplicated. They have the same genes twice on the same chromosome. Inversion, part of a chromosome could break off and then reattach upside down. And translocation, a piece of one chromosome could break off and attach to another chromosome. So all of those are abnormal. And remember, these chromosomes have the information for the cell to do stuff, but usually to make protein molecules. So if the genes are messed up, then the cell cannot make the proper proteins. And a lot of genetic abnormalities, genetic diseases that people have are because of there's something wrong with a gene. And so when the cell tries to make a protein, usually an enzyme that's required to do some kind of chemical reaction, if the cells can't make that enzyme, your body can't do that chemical reaction, there could be serious problems. Okay, so here's diagrams showing a deletion. This section labeled A is just broken off. Now it's missing. The duplication, the part labeled DE here, it appears twice. It's been duplicated by accident. Inversion, yes, the C and D part has been flipped upside down, so now it's reversed. And translocation, these pieces switched, and this is not the same as crossing over. I mean, this is what happens with crossing over. If these were both number one chromosomes, that would just be crossing over. But if this is a number one chromosome and this is a number 7 chromosome, that's not supposed to happen. That's that would be a translocation. Okay, some examples of syndromes caused by these kind of chromosome structural abnormalities. These have things like Williams syndrome, chromosome number 7 is missing a piece. I can't remember what the symptoms of this syndrome are. This Craigie-Shaft syndrome, it's called Cass-Cry. This is French, I think French for Cass-Cry. But a piece of chromosome 5, number 5, is missing. And it's called Cass-Cry because babies who have this, when they cry, they sound like a cat meowing. And then Down syndrome, of course, we said most cases of Down syndrome are caused by 3 number 21 chromosomes, but a small percent are caused by the translocation between number 21 chromosome and number 14 chromosome. So a piece of one very often attaches to the other. Translocations can cause some kinds of cancers, and also schizophrenia is known to be at least partly genetic. It seems to run in families. I guess they have located, scientists have found what gene on which chromosome is involved. Chronic myelogenous leukemia, that's a form of cancer. Burkitt lymphoma, that's another kind of cancer of the lymphatic system. Practice question again, comparing and contrasting meiosis and mitosis. And some videos if you want to look at them. Might help. Okay, any questions on chapter 7? Let's move on to this chapter, which this genetics is it can be it's pretty interesting. I hope it is to you. So genetics, a study of heredity. So how some characteristics are passed on from parents to their offspring by passing on genes. So some terminology, some new words we need to learn. The word trait just means maybe in this case it means a variation in the form or physical appearance of a person. And when we're talking about genetics, these are heritable. These are characteristics that are due to genes that are passed on from parents to offspring. This word allele, I don't think we've seen this word before in this class, but alleles are just it says alternate forms of a gene. So, for example, we have genes for eye color, and so if let's say you have two number 1 chromosomes. If the gene for eye color is located here on the number 1 chromosomes, you'll have 2 of them, one on each number 1 chromosome. So those are your eye color genes, but they could be different. One could be for blue eyes and one could be for brown eyes, or they could both be for blue eyes or both for brown eyes. But so we would call those alleles. So there's a blue eye color allele and a brown eye color allele. those are both eye-colored genes. So that's what it means by alternate forms of a gene. So that's what determines your traits, your characteristics, or the alleles that you have for each gene, each type of gene. Because you have pairs of chromosomes, you have 2 alleles for each gene, except on the X and Y. The X and Y chromosome, if you're a male, you have 1 X chromosome and 1 Y chromosome. The kind of genes that are on your X chromosome are not the same as the kind that are on your Y chromosome. So you only have 1 of each gene that's on your Y chromosome and 1 of each gene that's on your X chromosome if you're a male. Okay, this word locus in genetics just means where on the gene or where on a chromosome a gene is located. So this location on this chromosome where this eye color gene is, that's the locus for that gene on that chromosome. So both eye color genes are the same locus on the number 1 chromosome. This word genotype just means which alleles a person has. So if this is we use a capital B for brown eyes and a little b for blue eyes. So if this is what you have 1 big B and 1 little b, that's your genotype for eye color. If they were both big B's, that would be your genotype. And the word genome, that's just all of the genetic information in your cells. So which alleles you have for every kind of gene is your genome. Okay, human genome. Remember we're talking about DNA molecules, which are those 4 letters, A, T, C, and G. In a human genome, so in all your 23 pairs of chromosomes, there's 3 billion base pairs, or 3 billion pairs of letters. And there's more than 20,000 genes in a human genome. So if you divide 3 billion by 20,000, each gene, on average, is 150,000 letters long. 150,000 of those A, T, C, and G in one gene. So that's a lot of information in a very small, in every cell of your body, all packed in there. I read once there's some scientists in England figured out, or they figured out a way of converting human language into DNA. Because these were biologists and they were talking about how it's getting to be a problem to store all the data that they generate through their research, storing it on computer disks and stuff, and they thought, what if we would store it as DNA? I wonder if that would work. And they actually they took all of Shakespeare's sonnets. I'm not even sure what a sonnet is, but all of Shakespeare's sonnets, and they converted them into DNA code. And then they actually have those DNA molecules made by a DNA lab. And in all of that information, all of Shakespeare's sonnets, it was such a tiny amount of DNA in the bottom of a test tube, they could hardly see it. And then so they said, oh wow, this is really an efficient way of storing a lot of information in small space. And so they calculated, they figured that if we would take all of the information that has ever been written by all the humans that have ever lived, everything, and convert it into DNA molecules, store all that information in DNA. That amount of DNA would fit in the back of a small station wagon. But so that's, if you did that on computer desks or any kind of electronic computer technology, it would be way more space than that. So, and this DNA is something nature came up with billions of years ago, and it's way more efficient than the most advanced computer technology that you can even imagine. But the problem is with that is it'd be very expensive. It's very expensive to have DNA molecules made. So probably won't actually do that for storing information. Okay, I mentioned these words before briefly, an autosome. Autosomes are just the chromosomes that are not the X and Y chromosomes. So the first 22 pairs of chromosomes we call them autosomes or autosomal chromosomes. They're just the ones that are not the sex chromosomes. And so autosomal genes are just genes that are on those 22 pairs of chromosomes, not the X and Y. Genes that are on the X and Y chromosome, we call them X-linked or Y-linked or just sex-linked chromosome or sex-linked genes. And we'll see how traits are genetic traits are inherited. We can predict. We know the eye color of two parents. We can sometimes predict what color their kids will be, their kids' eyes will be, and because we know the pattern of inheritance. But the pattern of inheritance on genes on the first 22 pairs of chromosomes is different from the genes that are on the X and Y chromosome. So it's a little more complicated with X and Y because females have two X's and males have an X and a Y. So if you're a male, you got your Y chromosome from your father, always, because your mother didn't have one. But you get your X chromosome from your mother, and she had two, so you get one or the other. So it's a little bit different from the autosomal, but we can still make predictions. You know how, even with X and Y linked traits. Color blindness is one of these. It's on the X chromosome. Okay, in this PowerPoint, most of what we're going to be talking about is traits that are on the autosomal chromosomes. So we don't say specifically that this gene is on the X or Y chromosome. Just assume that it's not. It's on one of the other 22. And we'll talk about the X and Y chromosomes toward the end of the chapter. Okay. Many traits are passed on through a pattern of inheritance called complete dominance. And this is probably the one that you have heard about if you know anything about genetics. You've probably heard of dominant and recessive genes. That's what this is. Complete dominance means that if there's two alleles for a gene, one of them is dominant and the other is recessive. So, for example, that's what this eye color is. And so we use the capital letter for the dominant allele and the lowercase letter for the recessive one. Dominant and recessive means that if you have one of each, the dominant one is the trait that you'll have. And so this dominant over the recessive one. And so if this is your eye color genotype, you have one big B for brown and one little b, which is for blue, your eyes will be brown, not blue. That's what dominant, complete dominance means. Not all traits are like that. In many traits, there's two alleles, but neither of them is dominant over the other. We'll talk about that kind of pattern later. An example, this is not in your textbook, but this came from another textbook that we we use for general biology. But the case of earlobes is an example. The shape of earlobes is a genetic trait. So people have people's earlobes can be either be attached like this, which means the earlobe is attached to the side of the head all the way at the bottom of the earlobe. But free earlobes means they hang down a little bit below where they're attached. And that's determined by a pair of alleles that we call big E and little e. And big E is for free earlobes. Little e is for attached earlobes. So if you have one of each, you have the free earlobes. Just like if you have a big B and little b for eye color, you'll have the whichever is dominant. It's just the brown eye allele. Some more terms here, genetics terms. Homozygous. Homo means same. So if both of your alleles for a gene are the same, like if for eye color, if both of yours were big B, then you're homozygous. If both of them would be little b, then you're homozygous. So if you have both of them are the same and they're the dominant one, then we say you're homozygous dominant for that gene or that trait. If they're both the recessive one, then we say you're homozygous recessive. If they're different, hetero means different, right? So if you're heterozygous, that means you just have one of each. Your two alleles for a gene are different. So if you were a big B, you had a big B and a little b, you'd be heterozygous. So this is referring to genotype, which alleles you have. But the word phenotype means what is the actual trait that you have? What's the actual physical trait? So if your genotype is, if your eye color genotype is big B, then your eye color phenotype is brown eyes. In the case of genetic traits, your phenotype is determined by your genotype. Your genotype determines what your phenotype will be. Some phenotypes are not genetic. Some phenotypes, like if you how much muscle mass you have, it could be partly genetic, but also largely just about how much you exercise, how much you lift weights, and when you bulk up, get a lot of muscle mass. That's not because of your genotype. That's because of something that you did or something that happened to you during your life, something environmental. This term cross in genetics, a cross just means two parents make babies, make offspring. So if we would say if we cross a parent with this genotype with a parent who has this genotype, what would their offspring genotype be? And we'll see how we can predict that in a minute. Okay, so if a person has the trait from the recessive allele, then we know that person's genotype has to be homozygous recessive. So that means in the case of eye color, if I look at you and I see that you have blue eyes, I know what your eye color genotype is. It has to be this. You don't have any big B, because remember, big B is dominant, and that's for brown. So if you had a big B, you wouldn't have blue eyes. They'd be brown. So the only way you have blue eyes is if this is your genotype. If I look at you and I see you have brown eyes, all I know is that you have at least one big B. So your genotype could be this, or it could be this. Could have two big B's, or you could have a big B and a little b. Your eye color would be brown either way, because the big B is dominant. Maybe if I knew what color your parents' eyes were, I could figure out if you are big B or big B, little b. Or if I knew what color your kids' eyes were, that might help me figure out. Okay, but we've already said this. Kind of what I've been sketching on the board, but you have 2 of every chromosome, and you have the same genes and the same location or locus on both. So if there's an eye color gene here on one, there will be one eye color gene same place on the other one. They could be the same, or they could be different versions, different alleles for that gene. When a chromosome is duplicated in S phase of interphase, so the cell's going to divide, then those sister chromatids will have identical alleles on them. They're copies. And until maybe crossing over would happen during meiosis, then they could swap parts. Okay, again, Punnett square. Did you guys learn about Punnett squares in a previous biology? Okay, it's kind of fun, isn't it, to do these? But I don't know if this is even in your textbook, this new textbook we're using. But this slide is from a different textbook. But Punnett square is just a tool that is used in genetics for predicting what will be the genotypes of the kids, the children, if we know the genotypes of those parents. And so, well, so here's the 2 parents in this example, and we know they both have 3 earlobes, and we know that their genotype, they both have the same genotype, big E, little e. They're both heterozygous for earlobe genes. And so when this was the father here, when he makes sperm, about half of his sperm will have a big E, and the other half will have a little e. Remember, sperm are haploid, just have one set of chromosomes. So each sperm has only one of those earlobe genes, not 2. So when meiosis happens and the chromosomes separate into the daughter cells, the chromosome with a big E goes into one, a chromosome with a little e goes in the other, and so we end up with half the sperm have a big E, half have a little e. Same with the mother. When she makes eggs, half of her eggs will have a big E, and half will have a little e. They have one or the other. So if this is a Punnett square. It's just a little diagram. We don't typically have pictures in it. We just do the letters. So these symbols here, this means this circle with a cross under it means female. The circle with an arrow sticking out is male. So this would be the father here, and this would be the mother across the top. Could do it the other way. This could be mother and this could be father. It wouldn't matter. You get the same result. But these are the alleles in the eggs that this mother produces. Half of her eggs will have a big E, half will have a little e. Same with the father. Half of the sperm will have a big E, half will have a little e. If this mother's genotype was big E, then we put a big E here and a big E here. Just whatever their genotype is, you just put one letter here and the other letter here. So then you just combine the rows and columns. So big E here, big E here, little e here, and big E little e here. So these are the possible genotypes of the kids. That if these two had one child, it could be any of these genotypes. So even though they're both big E little e, they could have a kid who's big E big E. They could have a kid who's big E little e. They could have a kid who's little e little e. These are the possible genotypes of their kids. So big E, so 3 earlobes is big E. So if this was their kid's genotype, it had 3 earlobes. This would also be 3 earlobes, but this one, little e, is for attached earlobes. And so if this was a kid's genotype, it would have attached earlobes. Even though both parents had 3 earlobes, they could have a kid with attached earlobes. So which is more likely? Which genotype is more likely? How do you tell? You can count. One of these, 2 of these, 1 of these. So so there's 4 boxes here. 1 out of 4 is big E. 2 out of 4, or half, is big E little e. 1 out of 4 is little e little e. So we can predict there's a 1 in 4 chance that their child is going to be big E big E. There's a 2 in 4, or 1 out of 2, 50% chance that child will be big E little e, and 1 out of 4 chance that the child will be little e little e. We call that the genotype ratios, 1 to 2 to 4. So 1 big E, the 2 little e, or big E little e, the 1 little e. Another way of saying the same thing is 1/4, 1/2, 1/4, or 25%, 50%, 25%. Just different ways of saying the same thing. This genotype ratio. So what's the phenotype ratio? So what's the probability that their child will have 3 or attached earlobes like this? Just 1 out of 4, right? This is the only one of these 4 that is attached. The other would be 3 earlobes. So there's a 75% chance that their child will have attached earlobes. Only 1 in 4 chance that it will have 3 out of 4 that it'll be free. 1 out of 4, 25% that it will have attached earlobes. So that's the phenotype ratio: 3 free to 1 attached. For 75% free, 25% attached. These are just probabilities. This doesn't mean that if they have 4 kids, exactly 1 of them is going to have attached earlobes and exactly 3 are going to have 3 earlobes. Just like whether your kid is male or female, it's about 50% chance, right? Either way. But does that mean that if you have 6 kids, you're going to have exactly 3 males and 3 females? I had an aunt and uncle who had 7 kids. They were all girls. It's very unlikely. So these are just probabilities. But if in a Punnett square, sometimes you can find out that if these were all big E or big E little e, then we could say there's a 100% chance that child's going to have 3 earlobes. It's impossible to have attached earlobes. So sometimes we can say for sure they'll all be this, or none of them will be this. Sometimes we just can give percentages. This can be really important if there's a genetic disease that runs in your family, and you're trying to decide, should I have kids? What's the probability that if I have kids, the child's going to have that disease? And we've seen here that two parents that neither of them have attached earlobes, they could have a child with attached earlobes. What if there's a disease that runs in your family and your husband's or wife's family? Neither of you have it, but you know it runs in your family. Is it possible that even though neither of you have it, your child could have it like this? You want to know that before you decide to have kids. So there are people called, there's a profession called genetic counseling. People, their job is doing this. Couples who considering having kids, they know something runs in their family, one in one of their families, or maybe both, and they want to know, should we have kids? We don't want to have a child that's going to have some horrible disease. So they would want to know, what are the odds? Or is it even possible that it could happen? So let's do a Punnett square problem. If I said this is the mother, and we use an X for cross. That just means these two parents have children together. So what would their eye color of their kids be? You do Punnett square. So just take the letters, put the letters from one parent along the top and the other parent along the side. It doesn't matter which one you put on top or side, you'll get the same results either way. But then just cross and combine them. So in this case, all the possibilities are the same. So these two parents have a child. That child is going to be heterozygous, going to be big B, little b. Brown eyes. If they have 10 kids, they should all have brown eyes. What if they have a kid with blue eyes? That doesn't necessarily mean that mom was cheating with the mailman or something. It is possible for two blue-eyed parents to have a brown-eyed child or something, because these are the big B and little b are not the only genes that influence eye color. There's another pair of genes that we call them. We use the letter G for them, big G and little g. So it's actually a combination of your B genes and your G genes together that determine your eye color. So it's not just as simple as just looking at the big B or little b. Okay, so need to talk about this guy Gregor Mendel, who was an Austrian monk in the 1800s, and he had training in math and science, and back in those days, monks had a lot of time on their hands, so a lot of them did scientific research, and so he just working by himself. No one really knew he was doing this. he was doing experiments for years, genetic experiments, mostly using pea plants. Um, you take two parent pea plants, cross them, get seeds, plant them, see what those plants look like, and he do that generation after generation after generation, and he kept close records of the characteristics, the traits in every generation of these pea plants. And so he was a guy who figured out all this, figured out these patterns of inheritance, and he didn't even, this was before we even knew what a chromosome was. Scientists hadn't even discovered chromosomes or genes yet, and yet he figured out the patterns of inheritance, even though he didn't know about genes or chromosomes. He figured out how we can predict probabilities, like we've just been talking about. And no one even knew that he did this until after he died. He had kept record of this, and he was a monk. And he didn't, publish his research or anything. And it wasn't until much later that people discovered, wow, this guy really figured out something important. And now he gets credit for this very important information that he figured out. So before Mendel, people had been breeding plants and animals for many, many years. They knew that if you wanted dogs or some kind of livestock, any domesticated animals, if you wanted to have certain characteristics in them, the best way you could get that would be to breed two parents who had that characteristic, and you were likely to get it in the offspring. And the more generations you would do that, the more you would improve that kind of trait. So if you wanted large cows, large cattle, then if every generation you breed the largest ones, then generation after generation they might just get bigger and bigger. But they didn't know why that was. They didn't know anything about genes. And it was thought that there was this blending. They call it the blend. Now they call it the blending theory. If you have two parents and they're different, then they produce offspring. Then the offspring are going to be intermediate or in between. So if according to that blending theory, if we would cross a brown-eyed parent and a blue-eyed parent, we should get kids with brownish blue eyes. But that's not true. And it turned out this was wrong. It's blending theory. It's not that simple. We just saw how two parents with 3 earlobes could have a child with attached earlobes. There's no blending there. Anyway, Mendel's work proved or showed that blending theory isn't correct, and so he used this garden pea, this species Pisum sativum, for several reasons. They're easy to cultivate. You could grow them in pots really easily. Short generation time, just a few weeks. You plant a seed, a few weeks you got an adult plant that's producing seeds. So he could do a lot of generations in just 1 year. Get a lot of results. They produce many offspring, so many seeds. They're easy to self-pollinate or cross-pollinate. That means one plant could be both the mother and the father. That's what self-pollinate means. Or they could take separate plants. Make this one be the father and this one be the mother, by taking pollen from this one and putting it on the other one. And he used a bunch of different varieties of these peas, and by varieties it just means different combinations of characteristics, that genetic characteristics that he could cross in parents and see what the offspring look like. And so, and the traits that he focused on in these parents, in these plants that he would cross together, they were all traits that occur in only two forms, nothing in between. Like we know that in these plants that he used, well, there's a list of these kinds of traits. Yeah, like for example, the seeds are either round or wrinkled. There's an allele for round seeds and an allele for wrinkled seeds. So every one of these pea plants would have seeds that are either round or wrinkled. There's no in between, no kind of wrinkles, just one or the other. Yellow or green seeds, no in between, so one or the other. Purple or white flowers. The pods, the pea pods, either use the term inflated or constricted. So either they're the constricted kind, the pod is like as tight, so tight around the seeds that you can see the individual seeds through the pod or the shape of the seeds. The inflated kind is not tight like that. Pod color, yellow or green, and so on. So he would breed, maybe breed a plant that had round seeds with a plant that had wrinkled seeds, and then see what the offspring would have. And because these plants produce, each plant produces so many seeds, he would count. One plant might produce a few hundred seeds, and maybe some would be yellow and some would be green, and he would count them. He used these letters for the generations. The original, like the first generation that he would start with, he called that the P generation for parents. Those are the original parents, and then they would have offspring, and he would call that the F1 generation. And then he would cross some of these with others and get the next generation that he'd call the F2 generation, and then might do an F3 and F4 generation. And he would keep track, counting how many had this trait and how many had the other trait. And so when he found that in some cases if he would breed a tall plant with a short plant, then he would get, well, these are his actual numbers. He would get 787 tall plants, 277 short plants. And he'd do that with other traits, different pod shapes in the parents, and then he'd count how many of each were in the next generation. And something he noticed in all these numbers was it's not exactly, but approximately 3-to-1 ratio. So if you divide this number by this number, it's about 3. Divide this by this, it's close to 3. In every case, it's close to 3. So there's this 3-to-1 ratio. And he didn't know why that was. Nobody knew. But that ratio helped him figure out what's going on here. So he did these experiments called monohybrid. Monohybrid just means we're just focusing on one trait at a time, seed color, or tall or short, or wrinkled seeds or round seeds. Just one trait at a time. That's what it means by monohybrid. And so he'd start with plants that were what are described as true breeding. That means that if you take one of these plants and you self-pollinate it, which means the same plant is both the father and the mother, so this plant produces pollen. You put that pollen on the same plant's flowers, and then it produces seeds. And you do that generation after generation after generation. And those plants, when you do that, the trait is the same, generation after generation. So if you do this with a plant that has produces yellow seeds, and then the next generation will have all yellow seeds, do that again, the next generation, all yellow seeds. We call that a true breeding plant. And he didn't know why they were true breeding. Now we know that just means that plant is homozygous. So if this, I forget what, if you use it, yeah, big Y for yellow. So if this was a genotype of a plant, big Y, and big Y is for yellow, little y is for green. So if you would cross-pollinate this plant or self-pollinate this plant, it's the only kind of genotype in all of its offspring are also going to be big Y. There's no little y right there. So then if you cross this with itself, you're going to get big Y again. Same thing with little y plant. If you cross this plant with itself, you're going to get little y offspring, little y the next generation. So these are going to be yellow every generation. These are going to be green every generation. That means they're true breeding. We now know that's because they were homozygous. Mendel didn't even know what a gene was at that time. He just knew they were true breeding. So he took one of each. So he took a true breeding yellow plant and a true breeding green plant. And they're labeled here, big Y, little y. But remember, Mendel didn't know they were big Y, or little y. He didn't know what a gene was. He just knew they were true breeding yellow, true breeding green. He bred them together, crossed them together, and he got, the next generation, they were all yellow. Because they were all big Y, little y. Big Y is for yellow, so they were all yellow. He thought, what the heck's that mean? He didn't know anything about genes. He just knew he took a true breeding yellow plant and a true breeding green plant and cross them, and you get all yellow plants. Didn't seem to make sense. But then he would cross two of these together. These are two plants together, and then that's when you get that 3-to-1 ratio. 3 yellow to 1 green, or 300 yellow to 100 green in that F2 generation. And that was because, like we saw in the earlobes, we do a Punnett square and we get that same 3-to-1 ratio. 3 attached earlobes to 3 earlobes to 1 attached earlobe. That's why he was getting 3 yellow to 1 green. But if you cross 2 heterozygous parents, what these were, and you'll get 1 big Y, 2 big Y, and 1 little y. So that's the genotype ratio. The phenotype ratio is 3 yellow to 1 green. And so he was getting this 3-to-1 ratio in that F2 generation. no matter what he did, whether he did it with a seed color, wrinkled or smooth seeds, tall or short, he did the same 3-to-1 ratio in that F2 generation. And that's what those ratios, those numbers that he got. He worked backwards to figure out what was going on. He figured out that there were what he called factors, which we now know that they are alleles, but he just called them factors. That each parent has 2 factors, and when those factors, when the 2 parents come together, 1 factor from each parent is combined into each offspring, which will then have 2 factors, and that's what causes this pattern of inheritance. Much later, many years later, we figured out that those factors that he was talking about, those are genes on chromosomes. Even though he didn't know what a chromosome was when he was doing this. He knew nothing about DNA or genes, but he just from the math, he figured out what was going on. And we now know that the big Y is a dominant and the little y is recessive. He didn't know anything about that. He just called out one the expressed unit factor and the other the latent unit factor. Latent means sleeping. So what we now call the dominant gene, he called the expressed unit factor. What we now call the recessive one, he called it the latent unit factor. He just knew there was something in those plants that was being passed on. We call them factors. So this is what he called the law of segregation. Just that there are these factors that we now know are genes that separate when a parent reproduces. These factors or genes separate from each other, and each offspring can get one or the other. That's what the segregate means, to separate. And he just figured out there's something that he called factors that are separating from each parent, and one or the other of these factors goes into each offspring. Back up here a little. I skipped over something here called a test cross. A test cross. This is, for example, if you we've already learned, if an individual has the if an individual has brown eyes, for example, we know that individual could be big B or big B little b, right? We don't know which, just by looking at it. All we know is one or the other. You know, it's not little b, it's either big B or big B, little b. How could we find out? Well, we could do what's called a test cross. If you cross an unknown individual, that means an individual who has the dominant phenotype, but we don't know if that person is homozygous dominant or heterozygous. So we don't know if he's big B or big B, little b. We cross that individual with an individual with the homozygous recessive trait, and then we see what the offspring looks like. And that would help us determine what this individual's genotype is. So if we had a person with brown eyes And so we know that that person is either big B, or big B, little b, and we want to know. We could cross that person's with, let's just say this person's genotype is big B blank. We don't know what the other allele is, right? We cross this person with some who's little b, and we see what their kids look like. If they have, if they would have any kids who have blue eyes, would have a kid like this. What does that tell us about this?
Recessive.
Yeah, it has to be little b, right? This would have to be, if they have a kid with blue eyes, which would be little b, that tells us that this has to be little b. But if it was big B, then this kid gets one allele from each parent. This kid would have to have a big B. It would have to be big B, little b. We did that cross earlier, right? Big B crossed with little b. They would all be big B, little b. So we know if they have any kids, they're big B, little b. We know that this person's genotype is big B, little b, not big B, big B. If they had a kid that's brown eyes, then we still don't know for sure if that's a big B or little b. Could be either one. But if they had 20 kids and all of those kids had brown eyes, they were all big B, little b, we would probably be pretty sure that this is a big B. Because if this was a little b, out of 20 kids, they would probably have at least one that's big B, little b, or little b, little b. But we still wouldn't be absolutely sure. It would be possible for these two parents to have 20 kids that are all big B. So even though, let's do the Punnett square for that. Big B, little b, little b, little b. So big, little, little, little. So if this person's genotype was big B, little b, and we cross them with somebody little b, we would expect, according to this, we would expect half big B, and half little b, right? So if they had 20 kids, we would expect maybe about half of them would be brown eyes and half would be blue eyes. But if they had 20 kids and they were all brown eyes, that's pretty unlikely. But we still wouldn't be 100% sure that it's this. But if they had any kids that were little b, we would be 100% sure that this is little b. Is that right? Did I say that right? I'm getting myself confused now. But that's a test cross. You have an individual that has the dominant trait, but you're not sure the genotype. You cross that individual with someone with a recessive genotype, and you see what the offspring look like, and that'll help you decide. I can't imagine somebody doing that, going to the trouble of having kids just to find out what the genotype is. But people do that for in livestock and breeding crop plants. If they're doing serious selective breeding, of plants or animals. So they want to know what the genotype is that they're working with to start with. So they want to check across. Okay. Okay, law of segregation. Mendo also did dihybrid, and don't try to understand this. It's more complicated, but a dihybrid means he's focusing on two traits at once, like tall and short and seed color. And he's got that's what would be. There would be 4 letters. So this individual, big R, big Y, big Y. R is for round or wrinkled. So this would be wrinkled seeds and yellow seeds. This individual would be, this is brown seeds and yellow. This would be wrinkled and green seeds. And then you would cross these two. And see so there'd be lots of different possible combinations in the offspring. Could be big R, big Y, big Y, big R, big R, big Y, little y, and so on. And there's 16 possible or how many combinations? Well, here's what the Punnett square would look like for that. So instead of just being 2 by 2, it's 4 by 4. So there's end up with 16 offspring to count. And you get this 9-to-3 to 3-to-1 ratio of traits, or combinations of traits. And now this just told him that he got all these different combinations of traits in the offspring. That just told him that, for example, when what we now know as the genes or chromosomes, when the chromosomes separate in meiosis, the big Y and the big R, or this big Y and this big R don't always stay together. You could get a big Y and a little R, or a little Y and a big R, or a little y and a little r together. And that's that law of independent assortment, the independent assortment of chromosomes. The number 1 chromosomes separate independent of what the number 2 chromosomes do, independent of what the number 3 chromosomes do. So you can get all these different combinations of genotypes in the offspring. So that's what that 9-to-3 to 3-to-1 ratio told him that. If they stayed together, then if the yellow and round always stayed together, then you couldn't get a yellow and wrinkled in one plant, or you couldn't get a green and round together in one plant. But he got all combinations, so that told him that the chromosomes separate, or they assort independently during what we now know as meiosis. Okay, this is a good place to stop. We're out of time. So we are going to have an exam next week, right? Exam on chapters 4, 5, and 6.